Welcome back, everyone, to today's Explainer. So today, we're diving into a genuinely fascinating translation problem. We're going to look at exactly how we take a super messy, living biological process, stuff happening right inside a cell, and turn it into a highly structured, totally analyzable digital data format. Basically, we're going to see how modern computational biology turns actual life into a spreadsheet. Let's get into it.
All right, here's our roadmap for today. First, decoding gene expression. Second, from cells to sequencing. Third, the data structure explained. Fourth, the protein proxy assumption. And finally, scaling up to big data.
Okay, starting right off with Section 1, decoding gene expression. So you probably already know that biological information generally flows from DNA to mRNA and then finally to proteins. Well, for building our data structure, the absolute critical step we care about is that middle one, transcription. By observing the higher or lower levels of this mRNA, we're essentially capturing vital clues. I mean, these expression patterns are literally the key to telling a healthy cell apart from a diseased one, or, understanding how entirely different cell types behave.
Now, what's really cool is that both DNA and mRNA are basically just readable sequences of letters, because they're fundamentally just strings of characters, nucleotides, right? Modern experimental techniques can literally just read them like a book. This effectively digitizes the molecule. And honestly, this fundamental readability is the core mechanism that allows us to take physical biology and transform it into quantifiable data.
Moving on to Section 2, from cells to sequencing.
Okay, so to actually get our hands on this data, we can't just sequence mRNA directly. It's a bit too unstable. Instead, we have to use an intermediate step. We convert that mRNA back into complementary DNA, or cDNA for short, just to make the whole sequencing process possible.
All right, so here is the exact pipeline of how a physical cell's contents become digital info. First, after the mRNA becomes cDNA, it's broken down into these tiny little fragments. Those are then amplified into what we call a library, and finally sequenced by a machine. Once we have these newly sequenced fragments, which we usually call reads, we play a giant game of matching. We map them straight back to the original reference genome to see exactly which gene they originated from.
Which brings us to Section 3, the data structure explained. Now this is the main event. This matrix explicitly maps the biological world directly to the digital one. As you can see, every single row is a biological gene, and every column represents a physical tissue sample. Notice those integer counts in the middle, like that 8 for gene 3 under tumor sample 1? That simply means exactly 8 fragments of cDNA mapped perfectly back to gene 3 in that specific sample. Boom, that right there is our quantifiable read on gene expression.
And the real beauty of this standardized format is just its extreme versatility. It's almost magical how much complex science fits into such a simple grid. Literally, just by swapping out what those columns represent, you can answer totally different biological questions. We're talking tumor versus healthy tissue, drug exposed versus control, or even a CRISPR knockout versus wild-type cells. Through all of that, the underlying data structure remains exactly, elegantly the same.
Now I do need to give you a quick reality check here. You definitely don't just pop a sample into a sequencing machine and instantly get this perfectly clean spreadsheet out the other side. Not even close. It actually takes a massive amount of computational heavy lifting and rigorous quality control, a process known as low-level bioinformatics, to turn that raw experimental output into this neat little matrix. But for the sake of our analysis today, we're just going to go ahead and assume that this crucial processing step is already done.
So, what those columns actually represent depends entirely on the tech you're using. In standard bulk RNA-seq, a column is basically like a smoothie. It's the total mRNA from thousands of cells just mashed together. But in single-cell RNA-seq, every single column represents the exact mRNA contents of just one individual cell. Oh, and as a quick side note, older microarray tech actually gives us decimal values instead of those clean integer counts, but the matrix structure itself completely identical.
All right, Section 4, the protein proxy assumption.
Now, I really want you to think deeply about this one particular constraint. It's a bit of a puzzle. If proteins are the molecules doing all the actual heavy lifting in the cell, why in the world are our massive sequencing experiments entirely focused on measuring mRNA? Well, it basically comes down to technological limits. See, proteins are made of amino acids, not nucleic acids. And that means you simply cannot sequence them at the same massive, big data scale as DNA or RNA. You just can't. So we have to make an implicit assumption.
We treat the mRNA level as a roughly reliable proxy for the actual protein production. Now, is it perfectly true 100% of the time? Definitely not. But right now, it is absolutely our best big data workaround.
And that brings us to our final section, Section 5, Scaling Up to Big Data.
So, remember that neat little matrix we looked at earlier with just a few genes? Yeah, that was just a conceptual toy. In reality, a standard human gene expression dataset is going to have roughly 20,000 rows to cover all of our genes, and it's going to span hundreds, maybe thousands of sample columns. We are talking about incredibly dense matrices here, containing literally millions of individual data points. It's huge.
Which, of course, leaves us with the ultimate question, right? How do we actually analyze a matrix of 20,000 genes and hundreds of samples from first principles? Like, how do we comb through that massive pile of raw data to figure out real biological realities, like exactly which genes are driving a lung cancer tumor? Well, the answer lies in a super fascinating computational technique known as differential expression analysis. But how does that math actually work? Well, I'm going to leave you to ponder that question as your very next step in the incredible world of bioinformatics. Keep analyzing and stay focused.